While, but here's your next lesson on membrane permeability. Hopefully it's a short one. First, let's talk about what a selectively permeable membrane is and how the properties of phospholipids and the phospholipid bilayer make a membrane selectively permeable. So, plasma membranes, their function is they separate aqueous solutions from each other. Because membranes are selectively permeable, that means that they will only allow certain materials to pass and they'll prevent other materials from passing through them. And this is primarily due to the fact that the plasma membrane has that hydrophobic interior that we talked about in the last set of notes. To change this permeability, you can then have some embedded or stuck inside channel proteins or transport proteins that can change the permeability of these membranes. And then you can have membranes for the different organelles and cell compartments have different permeability by having different membrane proteins stuck in. So you could have a cell membrane that, for example, allows sodium ions to pass as long as you have a sodium channel in it. You could have another membrane that allows water to pass by having aquaporins, which are water transport channels. So let's talk about what can pass just a plain bilayer. If it's a small nonpolar molecule, it can pass freely. So here I have 3 different molecules. This is N2, which is nitrogen gas, O2, oxygen gas, and CO2, which is carbon dioxide. Notice that they can freely pass. They are hydrophobic and they are very small, so they can fit in the space between phospholipids. Hydrophilic substances, larger polar molecules and ions, cannot cross on their own. Instead, they need a little help. They need an embedded channel or a transport protein to get through. This is because they can't pass through those hydrocarbon tails of the phospholipids because they are polar, so they are more attracted to water, polar molecules, and those hydrophilic heads. So this, for instance, is a glucose molecule, but you could also have, say, an ion, positive or negative. Those would not be able to get through the hydrophobic layer and they would be bounced back. Now, small polar or uncharged molecules can pass through the membrane, but they do so very slowly because they are kind of not quite repelled by that hydrophobic layer, but they are going to be more attracted to the hydrophilic molecules in the watery solution. So here I have 2 examples. I have ammonia, which is this guy right here, and I have water, which, looks kind of like a Mickey Mouse head. You can see that some of this water is getting through, a small amount relative to the amount that's actually in this solution, and a small amount of ammonia is getting through, but a lot of it gets bounced back, which is what you're seeing here. So it might either bounce off or it might be more attracted to those hydrophilic heads of the phospholipids. That's why you need these channel proteins called aquaporins that are going to freely allow the passage of water, which we're going to talk about in much more detail when we talk about osmosis in another lesson. So now let's talk about the cell wall. The cell wall also is going to have an effect on whether things can get into and out of the cell because it's a barrier that's usually outside of the cell membrane. It is found in most forms of life except for animals, so bacteria, archaea, which if you haven't heard of, we will talk about those when we get to evolution and taxonomy. Fungi and plants all have cell walls. They are made of different combinations of macromolecules. So here we have 2 types of bacteria, and you'll notice they have different structures. So this one, you don't need to know this difference. Gram-negative bacteria have an outer membrane and an inner membrane and a layer of a molecule called peptidoglycans, which is a protein carbohydrate hybrid type of molecule. And then you have these gram-positives, which are going to have this type of acid as well as a peptidoglycan layers outside of their cell membrane. Here is a plant cell wall, and you'll notice it has a lot of cellulose fibers. It's got some glycans, which are carbohydrates that are going to link things, and then it also has pectin, which is a molecule you don't really need to know much about. And then fungi have different types of proteins. They have manoproteins, as well as glucans, which I assume are a type of carbohydrate, and chitin, which is definitely a type of carbohydrate. So you'll notice different organisms have different compositions to their cell walls, but what they have in common is usually they're more rigid and more permeable than a cell membrane is. So what does the cell wall do? It helps to maintain cell structure and function. It's an extra structural boundary. It does have selective permeability. So in plants and fungi, the cell wall is usually permeable to small molecules, some nutrients, but not to large molecules. And then its big role is it protects from something called osmotic lysis. And this is when a lot of water is entering the cell. So here you see in this picture a cell with the cell wall that's intact. Water can enter and the cell wall is not as stretchy or flexible as the cell membrane. So the cell won't pop once you have a lot of water entering. Instead, if you have here, this is a bacterial cell with a defective cell wall and water is entering by osmosis. And since it's got a defective cell wall, it can't protect itself from lysis and it pops. That's going to become really important when we talk about osmosis, especially in plant cells. All right, there we go.